Press the bell icon on the YouTube app and never miss another update.
Hi everyone. In the last video I discussed about menstrual cycle. Today I am going to talk about the process of fertilization. So you already know about the detailed structure of male reproductive system, the female reproductive system, how the sperms are produced in the testes and how the secondary oocyte is formed and released into the pelvic cavity. So you know about all these things in detail already. So now I am going to talk about fertilization. So what is fertilization? The fertilization is the process of fusion of haploid male and the female gamete to form a diploid zygote. So fertilization is what? It is the process of fusion of haploid male and the female gamete to form a diploid zygote is called as what? Fertilization. So what is the significance of fertilization? To restore the normal diploid set of chromosomes. In human beings, you know that the number of chromosomes are 46. Fertilization results in the restoring of the normal diploid set of chromosomes in human beings. Okay, now, so during fertilization what happens? The sperm, that is the male gamete, and the egg is the female gamete. So the sperm is haploid with 23 number of chromosomes. The egg is also haploid, no, it's also having 23 number of chromosomes. So during fertilization the sperm fuses with the egg. Okay, so that results in a single cell that is called as the zygote. So this is a diploid. Okay, so what is the number of chromosomes here? It is 46 number of chromosomes. So once the fusion happens, the fusion of gametes is also called as syngamy. Okay, so the fusion results in the formation of a diploid zygote. Okay, so the number of chromosomes is restored here, 46 number of chromosomes. Okay, so fertilization is the process of fusion of haploid male and the female gamete to produce a diploid zygote is called as what? The fertilization. Okay, where fertilization will occur in human beings? In human beings, the fertilization is internal. It occurs in the female reproductive tract. Okay, the fertilization will exactly happen in the ampulla region of the fallopian tube in the female reproductive tract. So you already know about the oviduct or fallopian tube. It is having three regions, infundibulum and then ampulla and then isthmus. So where the fertilization will occur in the female reproductive tract? In the female reproductive tract, the fertilization will occur in the ampullary region of the fallopian tube. Okay. Ampulla region of the fallopian tube, and these are all the steps involved in the process of fertilization. So let us see what are all the steps involved in the fertilization. So the release of egg, insemination, movement of the sperm, capacitation, acrosomal reaction, cortical reaction, sperm entry and the amphimexis. So these are all the events, the different steps involved in the process of fertilization. Okay, so we will discuss one by one in detail. So what is the first step? The release of egg. So it is not actually egg because it has not yet completed its second meiosis. No, it is the release of a secondary oocyte. So you already know about oogenesis process, right? So during oogenesis process, only one secondary oocyte is released from one of the two ovaries, right? So it is during the middle of the menstrual cycle, the LH hormone level will goes to a peak. That is what the LH surge. Okay, so it is due to LH surge the Graafian follicle will rupture. Okay, it's due to LH surge, increased increased level of LH hormone causes the rupturing of Graafian follicle. So that results in the release of a secondary oocyte from the ruptured Graafian follicle, that is called as what? Ovulation. So ovulation will occur on the middle of the menstrual cycle, that is during the 14th day of the, usually it occurs on the 14th day of the menstrual cycle. It is because of which hormone? LH hormone. Okay, so now during the middle of the menstrual cycle, only one secondary oocyte is released from one of the two ovaries. okay, from the ruptured Graafian follicle. Okay. So now the released secondary oocyte, the secondary oocyte is released into the pelvic cavity, and now the released secondary oocyte is picked up by the fimbria of the fallopian tube, and it is through the ostium the secondary oocyte will enter into the fallopian tube. Okay. So now it is in the region of infundibulum only. So now the secondary oocyte has to be carried to the region of, to the site of fertilization. The peristaltic contraction and along with the ciliary movement helps in carrying of the secondary oocyte to the site of fertilization, that is to the ampulla region of the fallopian tube. Okay. So once the secondary oocyte get into the fallopian tube, it is by the ciliary movement and the peristaltic contraction, the secondary oocyte is carried to the site of fertilization. So once the secondary oocyte is released, it is capable of living for only 24 hours. So the viability of the secondary oocyte is 1 day, that is 24 hours. Okay. So viability means what? It is the ability to fertilize. Okay. So it is the duration is 24 hours. So during this period only the sperm has to reach the egg, then only the fertilization will happens. If the sperms are unavailable during this period, then the fertilization will not happen and the secondary oocyte gets degenerated. Okay. This is what the first step, that is the release of secondary oocyte. And the next step is called as what? Insemination. So insemination means what? Insemination means the deposition of sperms into the female reproductive tract. So this happens at the time of sexual intercourse. So during sexual intercourse or the coitus, the male will ejaculate nearly 2.5 to 5 ml of semen into the vagina of the female. So the volume of semen in each ejaculation is 2.5 to 5 ml of semen. Okay. So this semen contains nearly 200 to 300 million sperms. Okay. So in a single ejaculation the number of sperms present are 200 to 300 million sperms. Okay. So out of 200 to 300 million sperms, the 60% of the sperm should have normal shape and the size, and 40% of them should show a vigorous motility. For normal fertilization to happen, out of the sperms released deposited in the female vagina, so the 60% of the sperm should have a normal shape and the size, and 40% of them should show a vigorous motility. Okay. Then see here the viability of the sperm. So viability of the sperm, here the viability of the secondary oocyte is 24 hours, no, whereas the viability of the sperm is 48 hours. So it is capable of, once it is deposited into the vagina, once it is ejaculated into the semen, the sperms are capable of living for 2 days. Okay. So its viability, its ability to fertilize with the egg is 48 hours. Okay. So once so that is what? The insemination. Insemination means what? It is the deposition of sperms into the vagina of the female at the time of sexual intercourse. Okay. So after sexual intercourse the sperms get deposited into the vagina of the female. Okay. So once they are deposited, the sperm start their travelling to reach the site of fertilization. So where fertilization happens, that is in the ampulla region of the that is fallopian tube. No, the sperms are released in the vagina of the female. So from there they have to reach the site of fertilization. Okay. So they start their travelling, that is through the cervix and through the uterus, and finally it is through the cervix and the uterus they finally reach the fallopian tube. Okay. So not all the sperm will reach the site of fertilization, because most of the sperms are destroyed in the female reproductive tract. It may be because of the acidic pH of the vagina. So the acidic pH of the acidic environment of the vagina kill most of the sperms. And what are all the phagocytes that are present in the female reproductive tract? So they engulf the sperms. Okay. So out of 200 to 300 million sperms which are deposited in the vagina of the female, only 1% of the sperm will enter into the uterus region. So of that, only 200 to 250 sperms are able to reach the site of uterus. So out of 200 to 300 million sperms, only 200 to 250 sperms are able to reach the site of fertilization. And because most of the sperms, they get destroyed by the acidic pH, that is the acidic environment of the vagina, and the engulfing of the sperms by the phagocytes what present in the female reproductive tract. Okay. So once they ejaculated into the vagina of the female, they start their traveling, no? So that is what the movement of sperms. So how they travel? What causes the movement of sperms in the female reproductive tract? Okay. See, the sperms are having flagella, right? The beating action of the flagella causes the movement of the sperm in the female reproductive tract, along with the prostaglandins. So what prostaglandins present in the seminal fluid, no, they causes the contraction of the smooth muscles of the uterus and as well as the fallopian tube. Okay, so it is due to the contraction of the uterus and as well as the fallopian tube, and the beating action of the flagella will carry the sperms from the vagina to the region of fallopian tube, ampulla region of the fallopian tube. So what causes the movement of the sperms? The contraction of uterus and the fallopian tube along with the beating action of the flagella, so helps in the movement of the sperm. So in the female genital tract, reproductive tract, the sperms will travel 3 millimeter per minute. So with the beating action of flagella and due to the contraction of uterus and the fallopian tube, the sperm will swim at a rate of 3 millimeter per minute. And the distance between the vagina and the site of fertilization is nearly 30 centimeter, and the sperm will swim at a rate of 3 millimeter per minute. Okay. So the next step. So after the movement of the sperm, the next step it is called as what? Capacitation. Next step is called as what? Capacitation. So even though the sperm reach the site of fertilization, even though the sperm reach the oocyte, they are unable to fertilize the ovum. Why? Because it is unable to fertilize the ovum for a duration of 7 hours. Why? Because during this 7 hours, the capacitation of the sperms will occur. So capacitation means what? See, the freshly ejaculated sperms, no, the freshly ejaculated sperms are unable to fertilize the ovum because they have to be made active. So the activation of the sperm is called as what? The capacitation. So what is capacitation? The activation of sperm. Okay, so capacitation means it is the final maturation of the sperm which enable the sperm to fertilize the egg. Okay, so that capacitation process requires nearly 5 to 7 hours. So the freshly ejaculated sperms are unable to fertilize the egg, okay, unless they are made active. Okay, so that duration is nearly about 5 to 7 hours. So what happens during capacitation? So on the membrane of the sperm head, some substances like cholesterol, glycoproteins, and the proteins get deposited. Okay, so the secretions of female reproductive tract will remove all the coating substances present on the membrane of the sperm head, okay, especially on the acrosome. So to prepare the plasma membrane of the sperm to penetrate into the egg. Okay, and this capacitation also causes that vigorous beating action of the flagella. Okay. So these are all the events that occur during capacitation, so which enables the sperm to penetrate the egg. Okay. So once the sperms are ejaculated into the female reproductive tract, they start traveling. Okay. So after traveling, so they are unable to fertilize the sperm before they are made active. So that activation of the sperm, it is called as what? Capacitation. Once the capacitation happens, so now the sperms are able to fertilize the egg. Okay. So then let us discuss the mechanism of fertilization. See, the mechanism of fertilization involves the acrosomal reaction, cortical reaction, sperm entry and the amphimixis. So, capacitation means what? Capacitation is the activation of the sperm. So, during activation what happens? The coating substances are removed and beating action of the flagella increases. Okay, the next process is the mechanism of fertilization. See, for fertilization to occur, the sperm has to penetrate the sperm has to enter the secondary oocyte. Okay, to enter into the secondary oocyte, it has to dissolve the membranes that covers the secondary oocyte. So, you know that this is the structure of a secondary oocyte. The secondary oocyte, it is having its own plasma membrane. Okay, that is the oolemma. So, this is again covered by a glycoproteinaceous layer that is called as the zona pellucida. Okay, the zona pellucida is surrounded by the corona radiata and some cells of cumulus oovarus will also be there. Okay, so the sperm, in order to enter the secondary oocyte, so these egg membranes has to be dissolved. Okay, so how these egg membranes are dissolved? It is with the help of the acrosome what present in the head region of the sperm. So you already know about the structure of human sperm, no? So that what acrosome which is present in the head region, which acts as it forms a cap-like structure around the nucleus, no? That is a modified Golgi complex. That acrosome contains sperm lysine enzymes. Okay, so these sperm lysine enzymes what present in the acrosome, they dissolve the egg membranes and help the sperm to enter into the egg. Okay, so what happens during acrosomal reaction? So I said only out of a million, only few sperms are able to reach the site of fertilization, and those sperms, these sperms now, they get added to the secondary oocyte. Okay, now the acrosomal cap, that acrosome will start releasing its sperm lysine enzymes, hydrolytic enzymes. So those hydrolytic enzymes, they are collectively called as sperm lysins. So the enzymes present in the acrosome are hyaluronidase and the corona penetrating enzymes and zona lysins. Okay, so what are the enzymes present in the acrosome? Hyaluronidase, corona penetrating enzymes and the zona lysins. Okay, so during acrosomal reactions, the sperms adhere to the secondary oocyte and the acrosome will release they start releasing its enzymes. Okay, now, so let us discuss the function of these enzymes. Hyaluronidase, what is the function of hyaluronidase? Okay, so this main function of this enzyme is to dissolve the hyaluronic acid, which acts as a cementing material between the corona cells and also the cells of cumulus oovarus. See, this is what the hyaluronic acid. So hyaluronic acid means what? It is a cementing substance which firmly binds the cells of corona radiata and cumulus oovarus. Okay, so this hyaluronidase enzyme now, it will act on the hyaluronic acid and it dissolve the hyaluronic acid. Okay, so that the cells get loosens. Okay, then later the corona penetrating enzymes. The corona penetrating enzymes will dissolve the cells of the corona radiata. Okay, so hyaluronidase enzyme will dissolve the hyaluronic acid, which is a cementing material between that is the cells of corona radiata and the cumulus oovarus, and later the cells of the corona radiata are dissolved by the enzyme that is corona penetrating enzyme. Okay, so the corona penetrating enzyme will dissolve the cells of corona radiata and the cumulus oovarus. Okay, now, so due to the dissolving of these layers, that is cells of corona radiata, now the zona pellucida get exposed. Okay, the zona pellucida layer get exposed. Now this zona pellucida, so the zona pellucida layer, it is having a ZP3 sperm receptors. So ZP3 sperm receptors are present in the zona pellucida. ZP3 sperm receptors, in the sense they are nothing but glycoprotein only, which acts as a sperm receptor. Okay. Now the sperm head that will bind with the ZP3 sperm receptor in the zona that is zona pellucida layer. Okay. So once it bind to the ZP3 sperm receptor, so that acrosome layer, that acrosomal reaction will begin. Okay, so during acrosomal reaction, what happens? The acrosomal covering gets degenerate. Okay, and its contents are exposed. Okay, so that contains what? Acrosin are also called as zona lysin enzymes. Okay, so these zona lysin enzyme will dissolve the zona pellucida layer. Okay, so these are all the sperm lysin enzymes, hyaluronidase, which dissolve the hyaluronic acid, and corona penetrating enzyme will dissolve the cells of corona radiata, so that the sperm will reach the zona pellucida layer, get exposed, and the sperm head bind with the ZP3 receptors in the zona pellucida, so that the acrosome covering gets degenerated and its content are released. Then what? Zona lysin are present in that content, so that digest the zona pellucida layer. Okay, so this is what? The acrosomal reaction. And once it happens, so now all the membranes that surrounds the secondary oocytes are dissolved. Now the plasma membrane of the sperm. See here, now the plasma membrane of the sperm fuses with the plasma membrane. This is what the plasma membrane of the this is the plasma membrane of the oocyte. This is the plasma membrane of the sperm. Okay, so after the acrosomal reaction, the plasma membrane of the sperm and the plasma membrane of the secondary oocyte fuses. Okay, so the fusion of the plasma membrane of the sperm and the secondary oocyte causes the depolarization of the egg membrane. Okay, now so the membrane potentiality of the egg membrane changes. Okay, so this depolarized membrane prevents polyspermy. So polyspermy means what? The entry of more than one sperm into the egg. Okay, so that is what polyspermy, and that polyspermy is prevented by the depolarization of the egg membrane. Okay, so this depolarized egg membrane prevents the entry of further sperm into the egg. That is, into the secondary oocyte. Okay, and so this acts as a fast black to polyspermy. So soon after the fusion of the sperm with the plasma membrane of the oocyte, so cortical reaction will occur. So this is the sixth step, that is the cortical reaction. So what happens in the cortical reaction? Some cortical granules are present beneath the plasma membrane of the secondary oocyte. So these cortical granules, so these cortical granules fuse with the plasma membrane of the oocyte and pour its cortical secretions, pour its secretion into the space which is present between the zona pellucida and the plasma membrane. See, this is what? The space. That space it is called as what? Pre-vitelline space. Okay. So once the sperm fuse with the plasma membrane of the egg, depolarization of the egg membrane occurs that prevent polyspermy. That is the fast black to check the polyspermy. And so soon after the fusion, cortical reaction will also take place. Cortical reaction means what? So these are all the cortical granules present beneath the plasma membrane. Okay. So these cortical granules, they fuse with the plasma membrane of the secondary oocyte and releases its contents into the space which is present between the zona pellucida. This is the zona pellucida layer and this is the plasma membrane. So what space present here? No, this is what? The pre-vitelline space. Okay. Now the cortical granules will secrete, it will pour their secretion into the pre-vitelline space that also contains some cortical enzymes. So those cortical enzymes will harden the zona pellucida. Okay. So hardening of zona pellucida again prevents polyspermy. Okay. This is the slow black to check the polyspermy. Okay. So the polyspermy is prevented by the depolarization of the egg membrane and also the hardening of the zona pellucida caused due to the cortical reaction. Okay. So it is because of this reason that is what because of depolarization and because of hardening of the zona pellucida, only one sperm is able to enter into the egg. Okay. So that is what the sperm entry. Okay. So at the point of contact with the sperm, a fertilization cone will develop in the oocyte. Okay. So a fertilization cone will develop in the oocyte. Right. So that it is the region that fertilization cone form. No? So it is in this region the sperm will enter into the egg. Okay. So soon after the point of contact with the sperm, the oocyte develop a fertilization cone to receive the sperm. So it is in the region of fertilization cone the sperm will enter into the secondary oocyte. Okay. So the entry of sperm into the secondary oocyte stimulate the secondary oocyte to complete its meiosis second. Okay, which get arrested at the metaphase second. Okay. So when the secondary oocyte will complete its second meiosis, it is after the entry of sperm into the secondary oocyte. Okay. So now the secondary oocyte, it completes its second meiosis, that results in the formation of a second polar body and a functional ovum. Okay. Now, so in the sperm, once the sperm it is already present in the ooplasm of the secondary oocyte, no? So it is having now it is having the nucleus and as well as the proximal centriole along with the other structures of the sperm. So all other structures of the sperm except the nucleus and the proximal centriole, all other structures of the sperm gets degenerate. Okay. So what is left is only the nucleus of the sperm and the proximal centriole. So that proximal centriole helps in the formation of spindle fiber at the time of cleavage, that is mitotic division of the fertilized egg. Okay. Now secondary polar body that is formed, no, that will also degenerate. Now the nucleus of the sperm and the nucleus of the egg will develop into a pronucleus. So they lost their nuclear membrane and their haploid set of chromosomes will fuses to form a diploid set of chromosomes, that is 23. Their fusion results in the 46 number of chromosomes. That process it is called as what? Amphimixis. So amphimixis means what? It is the process of fusion of the haploid male pronucleus with the female haploid female pronucleus is called as what? The amphimixis. Okay. Amphimixis is the fusion of male pronucleus with the female pronucleus. That process it is called as amphimixis. That amphimixis it results in the formation of a diploid zygote, what it is called? The fertilized egg. Okay. Diploid zygote. So that restores the normal diploid set of chromosomes in human beings. Understood? So this is what the steps which are involved in the fertilization process. Okay. So you will come to know the detailed information regarding the process of fertilization. Hope all of you understood. Thank you.